Okay, in this video I'm going to go through the rest of the question pack. Okay, so if you have your haemoglobin question pack at hand, we're going to review the questions. Okay, so starting with question 1. Okay, so question 1, the graph shows the oxygen haemoglobin dissociation curve for 3 species of fish. Species A lives in water containing a low partial pressure of oxygen. Species C, which lives in water with a high partial pressure of oxygen. And the oxygen haemoglobin dissociation curve for A is to the left of C. Explain the advantage of this to species A. So the first thing I kind of do when I look at these graphs is I start to sort of write some information on. So species A lives in a region where the oxygen concentration is lower because it says in the question it's got a lower partial pressure of oxygen where it lives. Which means that when you look at the graph at the same partial pressure, so at a comparable value, the graph tells you that species A's haemoglobin's got a higher saturation of oxygen. Therefore, it must have a higher affinity under those conditions for oxygen and load it more readily. And so it's that information that I'm going to be using to answer the question. Okay, so let me show you my answer. At the same partial pressure, so I always make that comparison, species A's haemoglobin has a higher affinity for oxygen and so binds more readily to the oxygen, becoming more saturated at the same partial pressure of oxygen. So I've basically described what I have observed from the graph, and that is my first mark point there. Okay, being more saturated was on the mark scheme, and having a higher affinity was on the mark scheme as well. Okay, so now we have to explain why it's an advantage, because we've only answered half of the question. As species A lives in low oxygen concentration, low oxygen conditions I've written, this ensures it is able to uptake as much oxygen as possible in that low partial pressure of oxygen to supply enough oxygen to the fish's tissues. Now, I've got the mark point there. I might have in some mark schemes missed it if I hadn't said for respiration. So always make sure you write that in. So you do have to answer in full in these. You can't just write in bullet points. You have to explain in full and try to use the information from the graph. Okay, so question part B. Okay, so question part B. Species A and B live in the same place, but B is more active. So as soon as the examiner tells you it's more active, you should be thinking oxygen dissociation curve shifting to the right, unloading being more important for that species' haemoglobin. Suggests an advantage to B of having an oxygen haemoglobin dissociation curve to the right of A. Okay, so my answer is B has haemoglobin with a lower affinity for oxygen. You can see that on the graph because the shift is to the right. If I go back to my graph, at the same partial pressure, comparing A and B, it's got a lower affinity. That's what I've said at the beginning of my answer. This is important because fish B, is more active and so needs more O2 to unload at the respiring tissues. Therefore, it needs haemoglobin with a lower affinity than A at the same partial pressure of oxygen. So its haemoglobin unloads more oxygen to the tissues because they're respiring more. So the mark point about respiring more, needing to unload more, is there, and the lower affinity mark point is there. But you can see I'm answering in full. I'm not just writing those two key points. Okay. One thing you need to bear in mind as well, it might not be clear from mark schemes, is all the time you must be talking about the hemoglobin of the organism. If you say species B has a lower affinity for oxygen, that's biologically incorrect. Okay, so species B's hemoglobin has a lower affinity for oxygen. That's a very common mistake people make, forgetting to talk specifically about the hemoglobin of the organism. Okay, so question 2. So there's quite a lot of STEM in this one. So I'm hoping you've gone through it just like me and you've noted all the important key terms. Okay, so red blood cell packed with full of hemoglobin when mature. It contains none of the organelles usually found in an animal cell. The nucleus, endoplasmic reticulum, mitochondria and ribosomes are all absent. So there is a list of things that would normally be in an animal cell that are not found in a red blood cell. More is known about the plasma membrane of a human red blood cell than about any other eukaryotic cell membrane. One reason for this is that the plasma membrane surrounding the red blood cell can be isolated without being contaminated by internal cell membranes. So remember, all the organelles, the nucleus, endoplasmic reticulum, mitochondria, all have their own membrane. And for most cells, that will mean that will contaminate any attempt to extract cell membrane from those cells. But when red blood cells don't have them, then that contamination won't be there. So red blood cell plasma membranes, or GOS, can be prepared by putting the cells in a dilute salt solution. Already I'm thinking about what water potential that will be. So this causes the cells to swell and burst, leaving only, so I put into the water, into the cells, so it must be a higher water potential, that salt solution, than the cells. So that leaves only the plasma membrane. Red blood cell GOS have been investigated and found to contain several different proteins. One of these proteins is spectrin. It is made up of long polypeptide chains which form a network on the inside of the membrane. Spectrin strengthens the membrane and is involved in maintaining the three-dimensional shape, that bioconcave shape we said was really important, of the red blood cell. Use the information in the passage and your own knowledge to answer the following. So A should be a nice easy one. You are doing a comparison not just of an animal cell now, but a red blood cell with a bacterial cell. So some of the things you would say about about animal cells, you can't say about red blood cells anymore because of the information that you've been given in the passage. So it would be fine to say that the red blood cell doesn't contain ribosomes. That's something that we know from the passage. Whereas we know bacteria contain 70 S ribosomes. So that would have got us a mark point. Another new thing that we've learned about the red blood cell is it has no nucleus, therefore it has no DNA. Whereas we know the bacteria cell has got that naked circular DNA. So there are some other things you could do as well, okay? But if you check out the mark scheme, these are the sort of best answers. Part B. Hemoglobin is a protein. Explain why a mature red blood cell cannot make hemoglobin. Well, again, this is from our cells topics. This is a nice synoptic question. Okay. Well, there's two things you could write about. It's lost its nucleus, so it doesn't have any DNA, which of course contains the code for the mRNA for protein synthesis. So it can't do the process called transcription. But also, it's got no endoplasmic reticulum or ribosomes, more importantly, which are the site of protein synthesis. So, or the site of translation, that part of protein synthesis. So that would mean it couldn't make any more proteins, it couldn't make any more hemoglobin. Part D. Just wondering where C's gone. Part D, I. Some people have red blood cells that do not contain spectrin. These red blood cells are spherical, so circle, like a sphere. They also burst more quickly when put in distilled water. Explain why more oxygen is uptaken by normal red blood cells. Well, a normal red blood cell has that bioconcave shape, giving them a larger surface area to volume ratio. Oxygen diffuses into them more quickly. Words that effect gives you a full tick. Okay. Next part. The decrease in the pH of blood plasma reduces the affinity of hemoglobin for oxygen. Explain how aerobic respiration in cells leads to a change in pH of the blood plasma. So we looked at this when we did the Bohr shift. So carbon dioxide is produced during respiration. It dissolves, forming an acid. You don't have to name the acid, but the idea that it's an acid, lowering the pH. What's the advantage of the tissue cells to this reduction in affinity of hemoglobin for oxygen when the pH decreases? Well, a decrease in pH causes a reduction in the affinity of hemoglobin for oxygen, so it is unloaded more readily at the respiring tissue. So that's my first mark. And why? Well, the tissue will then have more oxygen for respiration, so it can have a higher rate of respiration. Okay, another graph. Plenty of graphs in this one. Deer mice are small mammals. Straight away you should be thinking small mammals, large surface area to volume ratio, there's lots of heat across their surface, shifted to the right for the oxygen dissociation curve. They live in North America. One population lives at altitude. Oh, now you think, oh, isn't the examiner being mean? So now we know this little mammal lives in an area of low, or one population lives in an area of low oxygen concentration. The other doesn't, lives at low altitude. Less oxygen is available at high altitude, we knew that anyway. The graph shows the oxygen hemoglobin dissociation curve for the populations of deer mice. Again, can you see how I've processed my graph? I take a set value on the x-axis, which I call then my same partial pressure of oxygen, and then I go up and across and see what's happened to the saturation of each hemoglobin. And if you do that, the mice that live at altitude, you can see have a higher saturation. The hemoglobin, has a higher saturation at the same partial pressure as the other species, telling me it's got a higher affinity for oxygen and will load more readily. So that's what I used to do my answer. Explain the advantage for mice living at high altitude of having a dissociation curve, which is to the left of the curve of the low altitude mice. At high altitude, there is less oxygen available. They've told us that, so they're not going to give me a mark point for it. So it is an advantage to have hemoglobin that has a higher affinity for oxygen at the same partial pressure. That'll get me a mark point. It will load more readily, becoming more saturated, picking up enough oxygen for the tissues to use in respiration. That's what we want. Load more quickly, enough oxygen to use in respiration. You always need to put that bit on the end to get that second mark point. Suggest why it would be a disadvantage for the curve to be much further to the left. Now it's a bit of a compromise for our mice here, okay? If it was too far to the left, they'd have an issue in unloading the oxygen at the tissues. So, that should say two with two twos, too high an affinity for oxygen would result in hemoglobin that does not unload quickly at the respiring tissue site. So these tissues would have less oxygen for respiration. So if the mouse's hemoglobin had too high an affinity, that would mean it wouldn't unload quickly enough for the respiring tissue to get enough oxygen for respiration, is another way you could write that. Okay, now we've already reviewed 4. Okay, so we're on to 5. 5 is a bit of a calculation to start. Okay, so what you really need to be wary of whenever you are looking at biology and maths is that there's often something about unit conversion to be done. Okay, so obviously from this side here you can see how they've arrived at the rate calculation. So we've got 2 centimetres cubed per gram per hour, so that's our 20 centimetres cubed divided by the 10 grams to give us per gram. Alright, now we would just do exactly the same over here, but we need to make sure they're in the same units because we want to express it as centimetres per gram per hour. So convert the top one from kilograms to grams times by 1,000. The bottom one from decimeters cubed to centimetres cubed, which again is 1,000. So there's 1,000 centimetres cubed in 1 decimeter cubed, and then do the same calculation and you should get 0.105. 2 marks on that. It's really important to do that calculation in that way, converting the units first. Now they're trickier on the new spec, they might have only given you one of those in that way, so even if you didn't do it you'd still got the right answer if you like, without the conversion in this example, but you wouldn't on the new spec. They'd make it so it wouldn't happen. Okay, so and yet another graph question. Again, can you see straight away I do same partial pressure, what's happening to the hemoglobin of these 2 species, and then I always say at the same partial pressure, and I just read my graph. The hemoglobin of the shrew has a lower affinity for oxygen, which is what I can see because it's got a lower saturation. Okay, so go back to the question. It says the tissues of the shrew have a higher rate of oxygen consumption per gram. There is an advantage to the shrew in having hemoglobin with an oxygen and dissociation curve in the position shown, so it being further to the right. Okay. So at the same partial pressure, the hemoglobin of the shrew has a lower affinity for oxygen. I could possibly put in the evidence for that from the graph as well, shown on the graph, because it's got a lower saturation at the same partial pressure. So at tissues, when the oxygen dissociation, when the partial pressure of oxygen is at its lowest, the shrew's hemoglobin will unload oxygen more readily. So we want a lower affinity at low partial pressures, if you like, or the same partial pressure, meaning it will unload more readily at the tissues. So that's that first mark point, 2 mark points. Last mark point is always linking it to respiration. Okay, so I've said, and that oxygen is available for the greater respiration rate, which they talked about in the question. Okay, so question 6. The table shows factors which affect oxygen transport in the body tissues in different groups of animals. We've got reptiles, fish, amphibians, mammals. Suggest why the values for the mean rate of blood flow to the tissues are expressed per kilogram of body mass. This is a very common question in biology when you're thinking about data. So I've just started with because. Now our examiner said to start doing that. I find it quite hard, but she says just write at the beginning because, and then get down to answering rather than rewriting the whole question out again. So because these animals are all different sizes is the first mark point. So it allows comparison, those two. So it's not that it's easier to compare, it's the fact that they are different sizes. So unless you did it, you wouldn't be able to directly compare them. So they're all different sizes, so it allows comparison. Describe the relationship between the mean rate of blood flow and hemoglobin concentration. Well, I've done something that we'll start to do next year when we're looking for correlations. When we do Spearman's rank, when we're back in college, doing some stats at the end of the year. we will rank things and compare ranks. Now what I've done is I've ranked the blood flow smallest to highest, and I've done the same for the mean hemoglobin concentration. And if you look, fish was always the lowest for both, mammals the highest for both, and you can see the same pattern for the others. So that means I can clearly see a correlation between them, and that's what I've just been testing, starting to do a correlation coefficient. I'll show you the full one of those later in the year. So it's going to be a clear, there is an increase in blood flow as hemoglobin concentration increases. There's a positive correlation between those two factors. Question 7. Oh. Right, so question 7, another graph. Figure 1 shows the oxygen dissociation curve when the body is at, where's the end of that gone? Oh, rest. There you go. So at rest. Use figure 1 to describe how hemoglobin loads and unloads oxygen in the body. Okay, so we're using these two lines they've already put on really to do our description this time. So at the tissues where the partial pressure of oxygen is low, saturation is low, so there's a lower affinity. Our hemoglobin's got a lower affinity for oxygen. When the partial pressure is high, like in the lungs, we can see a high saturation telling us that hemoglobin's got a high affinity for oxygen. So it's just writing that in words really. So I've said in lung, I should have said in lungs, in the lungs, partial pressure of oxygen is high. Hemoglobin loads oxygen readily, becoming almost 100%. Because they've told me to use figure 1, can you see I'm starting to use data? Now this is where you might have lost a mark here. Can you see it's almost 100%? Whenever you're asked to use figure whatever, you need to start using data. So if there's data on the side, in this one you've got to quote it back. At the tissues, when the partial pressure of oxygen is low, hemoglobin unloads oxygen readily because it's got a lower affinity. Okay, so they're my 2 mark points. I probably needed more space to write a little bit more about affinity in that top one, but I did hit the mark point because I was using the data. During exercise, the oxygen hemoglobin dissociation curve moves to the right, as shown in figure 2. There we go, figure 2 to the right. So the same partial pressure of oxygen now, it's less saturated, it's got a lower affinity. Explain the advantage of this change. Explain the advantage of this change. I've said hemoglobin has a lower affinity for oxygen, therefore at tissues where there's a lower partial pressure of oxygen, it will unload oxygen more readily than at rest, meaning more oxygen will be delivered during exercise. Now the link in that last mark point is that more oxygen is needed during exercise because there's an increased respiration. So you don't get your mark point unless you talk about with exercise there's an increased demand for oxygen because of more respiration taking place. You could probably have worded it a bit better. Hemoglobin is a protein, so on 8, with a quaternary structure. What is meant by that? Oh, nice easy one. Made up of more than one polypeptide chain. Tick. How many has it got? 4. Tick. Nice one. Okay. C. Describe the relationship between the heme groups found on a hemoglobin molecule and the amount of oxygen they can carry. Okay, so each heme group binds to 1 oxygen, tick. There are 4 heme groups per hemoglobin, therefore each carries 4 oxygen molecules when fully saturated. Make sure you say when fully saturated. D. Explain how oxygen in a red blood cell is made available for respiration in active tissues. Tissues respire, and there's two effects to that, aren't there? Decreasing the partial pressure of oxygen and the pH. Okay, so the more respiration there is, the bigger this decrease is. So the bigger decrease in oxygen and the bigger decrease in the pH. And that would mean oxygen is unloaded from the haemoglobin as it has a lower affinity for oxygen under those conditions. And then it will diffuse into the cells from the red blood cell. So we want 1 mark for the change in conditions and a description of unloading more because it has a lower affinity in those conditions, and so there's more oxygen diffusing into the red blood cells for the second. That's the third, that's the second. So diffuses into the red blood cells is the last mark point on the end. Oof, nearly there now. Number 9. The graph shows the dissociation curve for haemoglobin from 2 different animals. The data for both animals was obtained whilst maintaining the same partial pressure of carbon dioxide. What's the difference in percentage saturation of the 2 haemoglobins at the partial pressure of oxygen, typical of active tissues indicated in the graph? Well, this is reading our graph, so we need to go from there, read off. So I had 20, 8, so it was 20 minus 8, that was 12%. Animal B is more active than animal A. Use the graph to explain how the haemoglobin of animal B allows for a greater activity level. So B has haemoglobin with a lower affinity for oxygen at the same partial pressure. So at low partial pressures of oxygen, it unloads more oxygen more readily, releasing more oxygen more quickly for the respiring cells to respire more. So this is for the increased activity. You have to have the word more. Okay. Final question. Question 10. Tubiflex worms are small, thin animals that live in water. They have no specialised gas exchange or circulatory system. Here's a picture of one. Name the process by which oxygen reaches the cells inside the body of the tubing worm. Of course, diffusion. Use the information provided to explain how 2 features of the body of the worm make it efficient for gas exchange. It's this making sure the so's here. So the fact that it was a thin organism, so there was a shorter diffusion distance, and the fact it was flat, so there was a large surface area to volume ratio for the diffusion. Okay, part C. Now this is new spec, and you can see the difference now. It's quite a lot more numbers, and we're expected to use the numbers within the answer a lot more. So most species of worms live at the bottom of the ponds where there's low oxygen. Pollution in the water can cause the partial pressure of oxygen to fall below 0.2 kilopascals. The graph shows the oxyhemoglobin dissociation curve for a species of worm found in a river polluted. The species of tubiflex worm in the graph has a 50% saturation when the partial pressure of oxygen is 0.08 kilopascals, so in other words below that polluted concentration. Explain how this enables this species to survive in water polluted with sewage. It's quite tricky, this one. So to get all the mark points, you've got to use the data. 50% is a high saturation for such a low oxygen partial pressure. It lives in conditions of 0.2 kilopascal. This means its haemoglobin must have a high affinity for oxygen at low partial pressures of oxygen, ensuring the haemoglobin loads oxygen readily enough even in 0.2 kilopascal conditions to provide enough oxygen, I've had to carry on at the bottom of the page, for respiration in the cells of the worm. So using my data to explain that there must be more, if you like, saturation than 50%, even at this low partial pressure, because of that property of a high affinity for oxygen. That's the first mark point. The second mark point is for linking to the respiration, making sure there's enough oxygen, readily enough oxygen ready, available for respiration in the worm, keeping it alive. Species of worm that live in ponds, lakes and rivers cannot survive in seawater. Use your knowledge of water potential to explain why. So we know it's got to be high water potential to low water potential. Well, seawater has lots of salt in, so it's going to have a lower water potential than the cells of the worm. It's your first mark point for realising that. So if you place the worm in seawater, water will leave its cells by osmosis, and that will kill it. So it's this idea that water will leave its cells down a water potential gradient or by osmosis, and the worm will die. Okay, so that finishes off the question pack. So we should now have question pack finished, student revision questions finished, Quizlet completed as well, and now you can do the spot test on the OneNote on haemoglobin. Brilliant, thank you.